Many proteins, when they are synthesized, fold into their three-dimensional shape spontaneously. But there are large, complex proteins that may fold into not the quite shape or may interact wrongly with large parts of one another, because many proteins are thousands of amino acids long. And those proteins may need assistance. They may need some help to fold into their correct structure. Chaperones are proteins, and their function is to help that folding process. And that's important because if a protein doesn't assume its functional structure, it can do other untoward things in the cell. And so there are processes that remove structures that are not folded properly. It takes a lot of energy to make a protein, and so the chaperones provide assistance to help capture that energy and not have to destroy the protein. Proteins come in a vast variety of functions, and we're going to talk about those functions momentarily. But ultimately, the function is derived from the shape of the protein, and the protein has to fold in order to carry out its target function. So examples: enzymes. Enzymes are cellular catalysts that carry out the myriad of reactions that happen in metabolism in the cell. They do literally thousands of different things, and surprisingly, they do it all at the same time in the same vessel. So they have great specificity. There are structural proteins that quite literally hold us together. There are regulatory proteins. There are many types of proteins, and we're going to talk about many of these types as we go through the semester. Interestingly, you can have a similar fold for a protein, such as the one I've shown here, but you can have distinct functions. There are families, for example, of binding proteins that recognize very different molecules. One family, for example, has components that recognize amino acids, that recognize sugars, that recognize ions, and recognize nucleotides. So the specificity has to do with the specific side chains and their orientation in the binding side. The inverse of that is you can have dissimilar folds. They don't look alike at all, that have similar functions, because the arrangement of amino acids in the active site, in the binding site or the catalytic site, is such that the reaction can be catalyzed or the target can be bound. We talked earlier about the next level of protein structure, if you will, and that is proteins that are oligomeric. Oligo, again, a Greek word, means few. So a few subunits. So you have dimers and tetramers and then much higher order oligomers. So if you have identical subunits, it is a protein that is termed homomeric. Different subunits, it's heteromeric. And so hemoglobin, shown here, is an example of a heteromeric tetramer. You have the blue subunits and the red subunits. And the blues are alike and the reds are alike. Now they are similar to one another, but they have differentiated over evolutionary time to have different sequences, even though they have similar functions. And oligomeric proteins are one way in which function can be regulated, and we'll talk a little bit more about that later. It turns out that proteins can assemble into much higher order structures than just tetramers or dimers or trimers. And one example of this is the viral capsids, the coverings that protect the genetic material in viruses from the environment. And in this case, there are 3 domains within a single protein that have different interactions and can form this interestingly symmetric viral coat. This happens to be hepatitis E. It is the cause of more than 50% of adult hepatitis in developing countries. And happily, the structure was determined by Dr. Tao in BCB. If you want more information, there's a link that you can find a YouTube that describes this structure determination in more detail. Proteins can be also, even in a single polypeptide, they can have multiple domains. And in fact, the proteins in higher organisms are very frequently more complex than those that are found in bacteria, which sometimes have only one or two domains. You can have these multiple domains that are linked together. This is a single polypeptide. It is synthesized as one long polymer, but they fold independently in these different domains and then carry out specific functions. Each of the domains is able to fold on its own, and you can in fact take these domains and mix them and match them and get a variety of different activities. Or you can alter one of the domains and keep the other two the same and get a different activity. Anytime you do a novel combination, you get a new set of functions that can be of use to the cell in a variety of ways. Interestingly, some proteins have regions that don't fold spontaneously, and these are called intrinsically disordered proteins. In fact, it took a very long time to discover these proteins because the nicely folded tertiary proteins structures that I have shown you, the monomers especially, purify really easily. Intrinsically disordered proteins behave very badly, and they're very difficult to purify. But at least some parts of many proteins, particularly in higher organisms, are not well folded, such as this disordered region shown in red. What this does is allow that disordered structure to respond to the proteins around it and form interactions with partners that induce the folding in this disordered region. And because it's disordered, you can have many different structures and therefore many different partners in carrying out its function. These regions are especially found in proteins that are involved in regulatory activities. They allow response to the environment because you can have different proteins interacting with the disordered region at different times in the cell, and when they interact, they have specific impacts on the function of the protein. They are most common in eukaryotes, although we're beginning to find more and more of them in prokaryotes as well, but a very significant percentage of eukaryotic proteins have some region that is intrinsically disordered. And interestingly, as we'll see later, these intrinsically disordered regions can also promote aggregation, which has health consequences and other things, and we will discuss that as we move along. So here is a video of intrinsic disorder. So you'll get a sense of, we've already talked about the movement, but this is a sequence that doesn't ultimately find a tertiary structure. It ultimately doesn't find the partners it needs to fold into a nicely ordered structure, and it remains intrinsically disordered. Some of these helices that form that you can see may be regions for partners to recognize, and if this happens to run into that partner, those regions may become fixed. So intrinsic disorder is incredibly useful because of its flexibility in the number of other proteins with which a protein will interact. So, can you think of ways that subunit interactions can regulate function? How might subunit interactions alter how a protein functions? And why are regulatory proteins more likely to exhibit disorder?